sweet voice again later in the evening or something and go over the extra stuff that I'm talking about with the study guide. But for right now, just get into it and I will show you what I'm talking about. Let's start with number one.
Mr. Jones, where is it?
Let me check your concept review. Go to your modules. I'll tell you where it's supposed to be, and then you tell me whether I'm lying to you or not. You go to modules, unit one. Yeah, at the bottom. Bottom. Unit one, vocab and concept review. It's the last thing in module one. Please tell me you see it. I'm not crazy. Everybody there, if you don't see it, please say something now before I start going over this and you guys are just sitting there listening to me.
I don't see it.
Yeah, I don't see it either.
Okay, you do see Unit 1 biochemistry. You're going all the way down. You see enzymes as the last major heading. You see at the very, very bottom, the last thing on that list says Unit 1 vocab and concept review. What's the last thing on your list under Unit 1?
Oh, I had to refresh the page. I see it now.
Okay. Well, it should have been there. All right. That's crazy. Fine. Open it up. I'll give you guys a second and I will open the Quizlet. I will be posting the link for this Quizlet at the bottom right beneath that. You will see it as soon as we are done here today. So that's why this is not an assignment. Your study guide, normally if we were in school, I would make that an assignment, but it's not because I want you to have the answers. I'm giving you the questions and the answers. All you have to do is look through them and memorize them. I don't
Mr.
need.
Jones, how are we supposed to fill it in if we can't type in it?
Make a copy. It should allow you to make your own copy and then you can do it. If you want, let me know later if you're still having an issue with that. If I need to, I'll just send you guys a link that will force you to make a copy. I think I just sent a shared link where you can view only. But if you go to file and say copy this, it should give you one that you own. Okay. Let's see. Unit 1, vocab, concept review. Hold on, let me get to it. It's at the bottom. All right, we will have this every time we have a unit. I have all of these Quizlets already made with your study guide, with the questions and answers. So we'll go through it together. Here it is. All right, number one. Number one, how are monomers and polymers different? Remember, this is called a concept review. We took the test questions and turned them into a concept. So you're not going to see this as the test question, but this question answers one or two, sometimes three things that could pop up on the test. So at some point, you're going to be asked about the monomer of the different macromolecules, and they might ask you what is different about the polymer. So monomer, you should know, is a single molecule. It's a brick, one brick. When I put a bunch of bricks together, I'm building something. That's your polymer, a string of monomers bonded together. For instance, the example we always give is glucose is a monomer. Cellulose is a polymer of glucose molecules. Cellulose is when I put a bunch of glucoses together. So is glycogen. All right, don't worry about writing these things in. Remember, I'm giving you the answers. You guys can type this in later or just use this to study. Just look at the question. Think about it for a second. That's why I'm pausing. Do you already know the answer or do you need me to give it to you? If you know these when we read them, You're in good shape. What type of organic compound is cellulose and what is its function? There's a reason why that's a question by itself, and that's why I went over it by itself with you. We just did this. What is it? It ends in OSE, so that's your hint right there. OSE means it's a sugar. If it's a sugar, it's a carb. And also the special thing about it. What is special about cellulose? It forms the plant cell wall, provides structure to a plant cell. Okay, again, number 3 is related to number 2. What type of organic compound is glucose? It ends in OSE. So you already know that. If you can know one of these, it will answer multiple questions. Glucose is the monomer of carbohydrates. All right, why is glucose so important to organisms? All right, one of you tell me, what do we use glucose for that is so important? Anybody? Come on, I know, be brave. Tell me what you think glucose is used for. You know this. You guys are just sleeping on me this morning. All right, that's fine. Fine. Make me do all the work. Glucose is important to organisms because it's our primary source of energy. It's the thing we burn first to get some energy. I wish you guys had some right now. Big old bowl of Fruit Loops. Primary energy source for all organisms is glucose. You know that because of that is so important, you can be guaranteed that will be a test question. List the polysaccharides commonly found in animals or animal cells. What did I just tell you a few minutes ago in the warm-up? Is the polysaccharide, when you put a bunch of glucoses together, the one that animal cells use for long-term storage of carbs. I gave you one for animals and one for plants. Anybody remember it? It starts with a G. Just yell it out. All right, let me put the chat over on another window because I know everybody wants to answer. That's sarcasm. Yes, glycogen, Jennifer. Thank you. Glycogen is the animal one. So expect on the test to see glycogen and cellulose because they are special and only work one way in an animal and in a plant. All right, list the polysaccharides found in plants or plant cells. And we just answered that. We gave the animal one, so the other one is going to be starch. Okay, it's referred to as starch, the storage polysaccharide commonly found in plants. So think glycogen for animal, think starch for plant. Those are the opposites of each other, animal and plant. And in the plant cell, one of those starches is cellulose, and you need to know what it is. So cellulose is not the answer for all polysaccharide in plants. The big umbrella word is starch and glycogen, but just no cellulose. You won't see both of those as an answer choice. They're not going to give you starch and cellulose, so you won't get confused. Okay, which organic compounds? There's 2 of them. 2 organic compounds give us energy. So if you can name the 4, which 2 give us energy? One is long term and one is short term. Come on, Jennifer, type it in. Most of these questions are answered by Michaela, Jennifer, and Maria. So one of you 3 got to remember. Which 2? There's carbs. But thank you very much, Parker. Way to step up. Lipids and carbs both give you energy. The short-term energy, the quick one, is carbs. The long-term stored energy, concentrated energy, is lipids. Expect to see that. All of these, we pared it down because there's a bank of 30-something questions. All I did for your test was I said grab random 25 questions and throw it together for each student. That's why each of you might have a slightly different test. These concept review questions cover everything that could be in those 37 questions. Some of those 37, they will ask the same thing 3 different ways. Now, if you get lucky and one of those things happens to be something you know very well, you got 3 questions in a row because you'll get that group together. But it's randomized, so you won't see exactly which ones. But if you know all of these concepts, multiple questions will be answered. What are the components of most lipid molecules? That was in our warm-up today. That's why I gave you that, just to make you remember. It's glycerol and fatty acids. Glycerol and fatty acids make up a lipid. Now, if it's multiple choice and you see the word fatty acids in there, that's probably going to be the thing that helps you out. A lot of you might forget the glycerol part. But if you see fatty acids and they're asking you about lipid, you can put those 2 together. Make a word association. All right, in your interactive slides, when we went to the lipid part, there was a picture of butter at the top of the slide. This next question asks, what are the negative health effects related to excess lipids in the body? If I have too much fat in my body, besides being overweight and having health symptoms because of that, what is also happening inside my body if I have excess lipid? And think of the butter. What is going on that might cause me a serious health problem? Anyone want to speak up, take a guess? Okay, what happens to people that eat way, way too much bacon their entire life? We're not talking about the weight problem. We're talking about what goes on with a serious health risk besides that. Nobody?
Their body starts making insulin?
Well, that's called diabetes now. Okay. That diabetes is not caused from eating too much fat. That's excess sugar. Yes, if you have too much iced tea is going to give you diabetes, but too much.
High cholesterol.
Yes, there you go. And cholesterol, if you know, cholesterol is one of your lipids. And what happens is it will turn sort of like the way the butter does. Think about bacon grease. I make bacon, I get clear oil at the bottom, but as soon as that pan cools off, it starts to get solid. Think about that happening in your blood vessels. You end up being at high risk for heart disease. That high cholesterol is also what will lead to heart disease. My heart will have to work harder to pump that blood because it's like you're pumping something thick like bacon grease at some point in your life. If it gets too bad, the blood can't circulate, your heart stops. So negative effects related to excess lipids is heart disease. All right, what is the function of nucleic acids? That's the easiest one of the 4. It only does one basic thing. So Na, nucleic acid, you know I'm talking about DNA and RNA. What's the function of DNA? Anybody remember the G word that you associate with DNA? What is it in charge of?
Genetic information.
Yes, both of you get credit for that. Genetic information. That's the one that you guys are going to get right because the others have all kinds of complex things that go with it. But DNA and RNA, genetic information. And I referred to it as the blueprint for your body. Your instruction manual for everything that happens in your body is held in that genetic information. You get half of it from your mom and half from your dad, but that controls everything for the rest of your life. Your looks, all the proteins that you make, everything that you express genetically is in that instruction manual. So that's its function. Nucleic acids carry the genetic information. Now it asks you, how does it do that? The second part of that is important. Remember the 4 bases for a DNA: A, T, G, and C. Those are known as nitrogenous bases because they have nitrogen in them. Those 4 bases, think of them as letters of the alphabet. How do we take those letters of the alphabet and make instructions out of them? Same way we do with our alphabet. The order of the letters. We take 20-something letters and we rearrange them in different ways and they have thousands and thousands of different meanings. Those 4 nitrogenous bases, the sequence, the way I put them together determines what their instruction says. So there's 2 of your macromolecules that depend on sequence. One is your nucleic acid. The other that has an alphabet to it is your protein, those amino acids. We have thousands of different proteins, but there are only 20 amino acids. It's almost like the alphabet that we have. 20 amino acids make thousands of proteins by the way you put them together. Same thing for nucleic acid. Try to make that association in your head, and that will help you remember. It's like letters of the alphabet. DNA and protein both do it the same way. Next question. What differentiates one protein from another? I just gave you that answer. The proteins are differentiated by the sequence of the amino acids. Okay, any of you remember what type of compound is hemoglobin? That was a warm-up question a few days ago. Which of the 4 macromolecules is hemoglobin, and what does it do for us? Why is it important? One of you future doctors, please tell me what of the 4, carb, lipid, protein, or nucleic acid, is hemoglobin? Let's start with that. Give me that half first.
Is it a protein?
Yes, it is. And also go back to my other rule I gave you. When in doubt, proteins do most stuff in the body. If you're stuck completely and you draw blank, guess protein. Now, what does it do? Hemoglobin, you got me the protein part. What does hemoglobin do for us? Seriously, if your hemoglobin is all messed up, what's going to happen to you?
It carries the oxygen.
Yes, you will die if without proper hemoglobin carrying your oxygen in your blood. That's why blood is red. Blood is not naturally red. Once it's carrying oxygen, it turns that reddish color. So hemoglobin carries the oxygen in the blood, and it's a protein. Next, if your body's in need of amino acids, all right, this is asking you if you understand what amino acids do. If your body's in need of amino acids, what type of food should you eat? So think about amino acids make what, and then what kind of food will give you that. Give me an example. First, tell me what amino acids do. Somebody tell me that part. They make. People, there's only 4 macromolecules on this test. It's one of 4 answers.
They make proteins.
Okay, so what am I going to eat to get some protein?
Like meat or nuts or something.
There you go. That's it. Something on the test is going to ask in a way so that you can tell, do you know what amino acids make and what kind of food will get you that? There'll be something on the test about that. All right, create an analogy for enzyme substrate relationship. I made you guys do a discussion question, a whole section on this. Give me a very, very simple answer. There's an analogy for how enzymes work. Anybody remember what it is? Okay, picture the little Pac-Man looking diagram. How do enzymes work, and what do we always say is the analogy for an enzyme? One of you knows this. Come on, Parker, give me another one. Pull that one out. We gave enzymes their own little lesson because of how important they are. You know you're going to see stuff about enzymes. Please make sure you answer the discussion question too. Some of you might miss out on. I didn't make it a whole lot of points because I know a bunch of you would forget about it completely. But the last thing on your list of assignments is a discussion question on how enzymes work. All right, I will tell you since nobody's coming up with the answer. Enzymes work like a lock and key. When you come to your house and you have a lock on the door, you have a key that has a specific shape. And if you don't have that key, you can't open that lock. So enzyme and substrate fit together like a lock and key. Shape matters, which relates to this next question. How does specificity relate to enzymes? That's just a fancy way of saying, what does the word specificity have to do with an enzyme. Each enzyme has a specific shape that fits only one substrate. Remember, it's enzyme fitting together like a puzzle with substrate. And one enzyme for one substrate. That shape matters. What can cause a protein enzyme to denature? We did this on our previous warm-up. The word denature means to change shape. What did we say can cause a protein or an enzyme to lose its shape? Anybody remember one of them? There's two things.
Temperature and was it pH?
Yes. Yes. Too much heat and too much acidity or basicity. Too much pH will knock it out of its optimum zone, and too much temperature will knock it out of its optimum zone. Now remember, you will see some graphs on the test. There's multiple questions with some graphs. Please look at them carefully and see what the graph is really showing you. Some of you are making assumptions when you take the test about what the graph says because you've seen a bunch of graphs and you answer the question incorrectly because of that. Enzymes work in a way where they reduce the activation energy needed for a chemical reaction. So most graphs, the ones you see most of the time, will show something that looks like a hill. There's the energy required. You have to get all the way up to the top of the hill before the reaction will happen. That is known as the activation energy, the top of the hill. But when it comes to enzymes, you also see other types of graphs. They might be showing you what is the optimum temperature for this thing to function. An enzyme will like to be heated up. It will increase its efficiency if it gets warmed up, but it hits a peak. When I get to that temperature right there, that's when that thing is moving as fast as it can. Now, if I keep heating it up, you see the curve start to come down. Now I'm damaging the enzyme. And when I get to a certain point, you'll see the hill come all the way back to the bottom. That's when I denatured it. It's no longer working. I just broke it. So look and see what graph they're asking you to interpret and make sure it makes sense to you. Do not assume based on the question. Then you look at the graph and you see it's showing you something a little bit different. All right, what are the defects of denaturation? I just did that. It changes the shape and the enzyme no longer works. What happens to the rate of an enzyme reaction if temperature increases? I just answered that too. Up to a certain point, you're increasing the rate of the enzyme reaction. Heating up things in science usually will increase your rate. I can take almost any reaction I'm trying to make and add some heat, and it will speed things up. All right, draw a nucleic acid and describe its components. I tried to show you this in the interactive slides that you had. There's a general shape for nucleic acid. They're not going to ask you to draw one on the test, obviously, but you need to know the three parts. And I made you label them in that interactive slide. There's three parts: a circle, a ring, a sugar ring like a hexagon, and then something off to the side, which is your A, T, G, and C bases. Circle, ring, base. Circle, ring, base. The circle is a phosphate. The ring is a sugar. And the base is that thing off to the side, A, T, G, and C. Sugar, phosphate, and base. Circle, hex, and box. All right, Chargaff's rule. We did this on the last warm-up. We're going to try and sample again. They're giving you a question here. This is a sample question. I think it's the actual question on the test, just to give you guys a hint. They're going to ask you this exact example. If I give you 30% G, all right, now if you have a piece of paper in front of you, or if you can picture this in your head, the four bases are A, T, G, and C. The two that go together are A and T, and then C and G. If I give you 30% G, that means that I automatically know how much C I have because they are equal. They go together, so they're equal. 30% G means 30% C. That leaves me with A and T. All four of these things have to add up to 100%. So now I just have to figure out how to split the rest of it between A and T. If I had 30G and 30C, that's 60%. That leaves me with 40. If I split between A and T, how much A or T do I have? Anybody?
20%.
Thank you. Yes. Simple math. So whatever they give you on the test, look at the percentage. Remember which bases go with which ones. The partner base always is equal. And then the others have to add up to 100%. So split the rest and you'll be able to tell what any one of those. They could have easily asked you, if I give you 30% G, how much A do I have? Or how much C do I have? They can ask it a bunch of ways. You just need to be able to figure out that math, and you got it. And this one is also very easy. You should be hoping you get this question on the test. If I get a DNA sequence, notice how it is laid out in threes. There's 3 letters, a little space. 3 letters, a little space. That's known as a codon. You don't need to know that right now. But each codon, all they're asking you to do is find the complement. The complement just means which bases go with those. So let's look at TAG, for instance. The complement of TAG, remembering Chargaff's rule, what goes with T? Anybody? Did you forget already? Which one goes with T?
Is it A?
Yes. So if I see a T, I'm going to write a little A right underneath that T, because that goes with it. The next letter in your 3 is A, and you just gave me the answer. What goes with A?
T.
Right? So I'm going to have an A, a T. Now, what's my third letter that goes under the G? Which one goes with that?
C.
Right. So on the test, all you do is go across for each 3 letters, put the one that goes with it, and it goes right underneath. And your answer choice is going to be the string that has the 3 that go with TAG, GGC, ACT, and TGG. You just have to remember the base pair rule, and complement means what goes with. What goes with it? If you're having a food and you say this food complements this food, what 2 things go together? Like fries and ketchup or whatever. The complement means the sets that go with those letters. And there's only 4 of them. And as long as you remember A, T, G, and C, easy answer to get. Last one. Compare and contrast the structures of DNA and RNA. On that interactive slide, again, you notice how many times I've referred back to that assignment. People who did it are definitely going to have an advantage. The interactive slide, when you got to nucleic acid, they gave you a picture of a DNA molecule and an RNA molecule, right one on top of the other. DNA is a ladder that's twisted. You have 2 sides to a ladder and a place to put your feet. If I twist that around, that's a DNA molecule. If I take that same ladder and rip it in half down the middle, so now I've got one thing to hold on to and half of a thing to put my foot on, that's an RNA molecule. So one is double-stranded, 2 sides. RNA is single-stranded, just 1 side. And the other difference is, remember I told you there's a base that's different. DNA has A, T, G, and C. RNA has a unique one called uracil, U, instead of a T. So it goes A, U, G, C. On the test, they might ask you, what do I have if I have uracil? I find uracil in a compound. What does it mean? The only place you find that is in RNA, so that's your answer. Okay, that's it. Now, quickly, I'm going to try to go through these, as many of these as I can with you. Go to the middle section of the module. Go back to Canvas, go to the middle section of the module, and find the QR codes. If I walk you through a few of these, let me know if you're getting it. I gave you an answer sheet. Click on the link that opens up the page with the board. And I'm going to look at the answer key while we do this so I can see, because I'm not going to obviously scan all these QR codes. But what I will do is go through with you the first few. I tried to give you like a couple to get you started, but I'm going to go as far as I can until we run out of time. So if you want, have your answer sheet ready in front of you, and we will just be going through. I would like you guys to give me some of the answers when these questions come up, and then you will fill it out on your sheet with me. In that 10 minutes, we should be able to get through half of these. Okay, now we will start from, I gave you a place to start. We'll go through from the beginning where I told you to start, because I started with question number 1. That'll make it easier for all of you to follow along. Let me find this sheet here. Okay. Here we go. I need this, and I also need. Okay, I started, look on the board, you'll see there's a card that has the answer lipid. It's a card that has the answer lipid. That is also the card that has the QR code for question number 1. So focus on the questions, people, and I'll just tell you, you'll know where to put the answer in on your sheet based on the question number. So just listen and see if you know the answers. Don't worry about the board. Just see if you can follow on the board so that when we get cut off here, you guys can continue. But if you found the card that says answer lipid, that QR code number 1 question is, what is the function of a carbohydrate? So one of you yell out or type, what do carbs do for us?
They provide energy.
Yeah. Okay. Yes. Both of you said something close enough to that. Yes. Quick energy. All right. So I would be looking for the card. If you guys can see the cards and you can move around. What I did was, when I made this, you can blow up the cards to a certain size, and then you can just use your mouse and drag it around so you can look at the different ones. The answer card that I'm looking for is used as the first source of energy. That's what carbs are, used as the first source of energy. Now, on your answer sheet under number 1, look for number 1 on your answer sheet and put used as first source of energy. So if you got that correct, the QR code on that card, when you scan it, it says question 2. What is the function of a lipid? All right, speed round, people. Yell it out. What do lipids do for us? Give me some of it. It does multiple things, remember? If it does multiple things, just give me one. That'll give us a hint on how to start. What card we're looking for. What do lipids do for us, people? Okay, yes. All right, Maria, you typed out a whole sentence for me. Long-term energy, insulation, part of the cell membrane. Okay, so that's easy. I'm looking for a card that has a bunch of stuff in the answer. The answer card that you would find says used for long-term energy, storage, insulation, and organs, and part of the cell membrane. So she typed out what was on that card. So on your answer sheet, under number 2, you will write out that long answer, or at least give me most of it and put ... Used for long-term energy, storage, etc. So if you found that card on the board, which Maria obviously did, you would scan the QR code, and it would tell you that number 3 is what is the function of a protein. So Maria, if you already found that one, you can start typing. But what do proteins do for us? Remember, they do the most stuff. But what is the function of a protein? Anybody? See, okay, Michaela got it this time. Structural components of an organism. I know she didn't pull that out of her butt, so obviously she found the card. Structural components of an organism: skin, hair, eyes, and nails. So if you found that card, which Michaela did, you would scan the code underneath that answer. So make sure on your answer sheet, function of proteins is what we just said. Makes up structural components of an organism. The code on that card is question number 4. What is the function of nucleic acid? See, we're all under functions right now. And nucleic acid was the one that multiple people gave me during the concept review. You guys know you're looking for genetic information. So, stores and carries genetic information. If you find that card on the board, you scan and it tells you examples of lipids include. So this one you will need some knowledge. Now, if you were doing this on your own, let's say you guys weren't talking as a group and you're doing this on your own and you can't remember, you would have to look through your notes or look it up online, whichever one you needed to. What are examples of lipids? That might not be something that pops into your head. But the answer was pretty easy. If you looked at the cards, it would have popped out at you. Yes, fats, oils, and waxes. You can remember lipid is fat. Fats, oils, and waxes are examples. So that's my next code. The question, so that's number 5. The answer is fats, oils, and waxes. That brings me to the next code, which is number 6, examples of carbs. That one, most of you should know on your own. You've known that before we even started the course. When I asked you guys to give me examples of carbs, you knew what you were looking for. But in this case, they're asking you for more basic info, not kinds of food. Carbs are sugars and starches. Sugars and starches. Sugar is the simple one. A bunch of sugars stuck together is a starch. Now, when you find that card on the board and you scan the code, the question that comes up on your phone would be, what are 2 types of nucleic acid? Easiest question on the board. Everybody knows that one. I don't even have to ask you to yell that out. The 2 nucleic acids are DNA and RNA. When you find that one, so that was number 7. 7 is DNA and RNA. Notice I'm pausing to let you guys, if you don't have it, you can put it in now. We're going to get through at least half of this together. Like I said, there's 20 and we're almost at number 10. So on the card that says DNA and RNA, when I scan my code, it says this is an example of a protein that helps to speed up reactions and lowers activation energy. This is why I told you this QR code thing is not a waste of time. This is all stuff that's in your concept review. And you know, because we just did it a few minutes ago, that the answer is an enzyme. Enzyme is a protein that speeds up reactions and lowers activation energy. So I'm looking for the card that simply says enzyme. Number 8 is enzyme. I scan that QR code and it asks me the monomer of nucleic acid. That's one of the easier monomers to remember because it sounds somewhat like the name of the thing. Nucleic acid's monomer is nucleotide. So now we're getting into monomers. You can see the questions have themes. I scan that QR code. So number 9 was nucleotide. And it gives me question 10. It's asking me the monomer of a lipid. Lipid monomer is what we did today. I made sure I put these warm-ups in the order I did because it's harder stuff to remember. The one we did today was glycerol and fatty acid. Monomer of lipid is glycerol and fatty acid. On the card, it says fatty acid and glycerol. So that's number 10. And we still have some time. So let's do 11. 11, if you scan it, the fatty acid and glycerol card gives you the monomer of a carbohydrate is. Monomer of carbohydrate, you know you're looking for glucose, but they're asking you for the technical term. Glucose is a monosaccharide. So process of elimination, when you get down to these cards, you would find, since we've gone through half of them, the cards that are left should give you a hint about what the answer is they're looking for. So the monosaccharide card, I scan that and it asks me the monomer of a protein. And boy, do we know that one because I've hit you over the head with it about 10 times just today. Monomer of a protein is amino acid. So that card, we can hit 13 and then I'll stop. You guys should be able to get through the rest on your own. Okay. The elements found, that's number 13 is the question that comes up. 12 was amino acid, 13. The elements found in protein are they're asking for the elements. That means your carbon, hydrogen, oxygen, that kind of thing. So does anybody remember from the Amoeba Sisters video what the elements are in a protein? Now, all of them have 3. All of your macromolecules have C, H, and O. But when you get to protein and nucleic acid, they have something more. So protein, the elements you need to remember are carbon, hydrogen, oxygen, and nitrogen. CHON, C-H-O-N. If you needed to look it up, you could have Googled that one. There's a couple of these that I could see you guys not remembering off the top of your head. So if you Googled that and found carbon, hydrogen, oxygen, and nitrogen, that's the answer to 13. And the next question there is the elements in a carb. That's the easy one. Carbs and lipids basically have the same elements, and it's the most basic ones. They're not special. It's just carbon, hydrogen, and oxygen. Now, on the card, the answer card, it also gives you the ratio. You guys didn't notice it before, but the formula for glucose is in a 1-to-1 ratio. It's C6H12O6. All of your carbs are in that ratio, 1 to 2 to 1. So find that card. All right, and we'll stop there. You guys are up to, that should give you question 15. So there's only a few left. The elements found in lipids are. The elements found in lipids are. And remember, I told you carbs and lipids had the same answer as far as the elements. So the way you distinguish between the two was the 1 to 2 to 1 ratio. That's why they gave you that. But it's the same answer as the one we just found. Carbon, hydrogen, and oxygen. All right, ask me now, people, if there was any way you think that you can't get through those last few. Because I think you guys can all finish that and get an easy 20 points that you went through this with me. All right, use office hours, people. If you need me for the weekend, before you get to your test, anything at all you need, jump in office hours or send me a message. I will answer you all weekend long. All right, otherwise, take your test, take your time, be careful. You only get one shot. Well, you guys might get more than one, but take your time. And I will talk to you on Tuesday. No class Monday, people. Don't show up here on Monday. Okay, I will talk to you guys later. Oh, come on. Okay, people, let me get my attendance up, and we got to keep moving. There's a lot to cover today to try to help you prepare for your test. That is our objective for today, making sure everybody knows what they need to know. Now, whether you remember it for your test or not, that's not on me. I'm just going to tell you what you need to know. I will give you full details because I told you it would be an advantage showing up today because half of your class probably won't. So anybody that's here, I'm going to make sure you have as much help as possible for the test. Make it worth your while. And we will also be going over the QR code scavenger hunt because that will also help us as far as studying goes. And I will show you, we'll be able to get at least through half of it. I will get you through enough of it so that you can finish it on your own. All right, there's your attendance. Now you have your attendance. There you go. 5 minutes, and then we will open up your warm-up. As soon as you check in, go to your module section, look in your warm-ups, and at the bottom you'll have one dated today, yesterday, 9/3. Open that up. We'll go through that because there's some stuff in there on the test. We will go through the study guide. I will give you the answers. The Quizlet that goes with it, I will post as soon as we get done today with class. And the QR code scavenger hunt also has things that will be on your test. I will go through from number 1. We will be able to get through. Last block, we got all the way down to question 15 out of 20. I will be giving you the answers. If we run out of time, you should be able to finish the